Microscopes are very important in microbiology because it is our primary tool for observing the myriad of microorganisms that are out there. So, as I mentioned previously, the human eye is good to about 0.2 millimeters. Anything under 0.2 millimeters, you're going to need some sort of lens, and for the vast majority of microorganisms, being much smaller than 0.2 millimeters, you're going to need to use a microscope with a series of lenses. The funny thing about microscopes, while they may seem like daunting tools, is that they work on pretty simple principles. And of all the scopes out there, they all kind of work on the same way. So most microscopes utilize a series of lenses, not just one, but several. And in order to illuminate the specimen, most microscopes have some sort of light source, be it a light bulb, a UV bulb, or in the case of electron microscopes, they utilize an energy source, an electron gun. But they all work on very similar principles. So there are three general types of microscopes out there. We're going to focus on light microscopes as we see them in lab, and they are the most common. You also have electron microscopes, which are not quite as common, but incredibly important for observing the smallest of specimens. And then newer microscopes, such as atomic force, allow us to visualize atoms and molecules, and more recently, chemical bonds. So similar diagram to what we've seen in Chapter 1, just kind of the grand scheme of microorganism size and which microscopes you need, depending on the size of your specimen. So in the top portion, the human eye is good, again, to about 0.2 millimeters. But the lens in your eye has limits. So beyond 0.2 millimeters, you need some kind of light microscope. Light microscopes allowing you to visualize small eukaryotic samples, human cells, even bacteria. But light microscopes have limits as well. So light microscopes are good to about just over 100 nanometers. Now, beyond that, you're going to need one of two types of electron microscopes, and we'll get into these later. One is more powerful than the other, but electron microscopes allow you to visualize bacteria even more up close. It's the only way for us to visualize viruses, but even they have limits. So, electron microscopes have limits, so that brings us to atomic force microscopes, which we'll see allow us to visualize atoms and molecules these days. But you have to remember, von Leeuwenhoek, the father of microorganisms, discovered them using one simple lens. Okay, so while microscopes are great for visualizing bacteria, you don't necessarily need them to see all microorganisms. Okay, so let's talk about the most common microscope that we deal with in microbiology, and the one that we get to see in our lab, bright field microscopes. So this is a type of compound microscope, meaning that it has two sets of lenses. There will be a carousel of objective lenses. So if you see here, I'm circling the objective lenses, of which they have increasing magnification of 4x, 10x, 40x, and 100x. And then you also have the ocular lenses as well. And those have a set magnification of 10x. So compound means when viewing a specimen, you have to multiply the power of the lenses you're looking at. So since you're looking through two lenses, in order to understand the total magnification of your specimen, it's a simple calculation. It's the power of whichever objective lens you're looking at, 4, 10, 40, or 100, times the power of the ocular lens, which is 10x. That never changes. So if you're looking at a specimen under the scanning objective, 4x, you would multiply that by the ocular lens, 10x, to get a total magnification of 40x. Total magnification. Very simple calculation. Since this is a bright field light microscope, It utilizes a light bulb. Okay, so the light bulb shines through and illuminates the specimen, and the light that bounces off the specimen is collected by the lenses, and it reaches your eyes, allowing you to perceive the object. The limit of our bright field microscopes is about 1000x, and even at 1000x, there's some limitations we'll have to deal with. So oil must be employed to help us resolve and focus on those objects at max magnification. Without the oil, we wouldn't be able to utilize our oil immersion objective lens. And the biggest drawback with light microscopes is that usually bacterial samples must be dead. So in lab, we're going to learn just how to do that, how to prepare bacteria on slides and how to kill them so that they stick to the slides. So that's a big limitation if you want to see living specimens. All right, so let's talk about what bright field microscopes do for us, and we're going to get to why do we need oil for that oil immersion lens. So microscopes have to be able to do two things for us. One is obvious. They magnify small objects, right? That's what lenses do. They make small objects look bigger. But the other thing that microscopes do is something that folks don't often realize or even talk about. Not only do they magnify small objects, they help you resolve small objects. So what does resolution mean? What does it mean to resolve small objects? Well, resolution is the ability of a lens, your eye included, to distinguish between two close together objects. So if you can see two objects that are close together, you're resolving them. Okay, but the thing is that every lens, including your eyes, have a limit of resolution. And I have a good example for you to think about or imagine to kind of help you grasp this. So imagine there are two people standing in front of you. Okay, you are resolving that there's two people there. But then ask them to start slowly backing up. Okay, eventually, those two close together people are going to get so far away from you, I would argue, that you can't tell them apart anymore. They're going to be so far, they're just going to look like a blurry spot. You've hit the limit of resolution for your eye. So, every lens helps you resolve objects, resolve that there's tiny close together objects. That's what your bright field microscopes do for you. They magnify bacteria, and they help you resolve that these tiny close together bacteria are there and you can tell them apart. But like any good lens, even your light microscopes have limits. So their resolving power is down to about 0.4 micrometers or 400 nanometers. Smaller than that, they can't really tell those small organisms apart from each other. So viruses, for instance, viruses are smaller than 400 nanometers. So a light microscope would be no good to you. So why do we have this limit of resolution? Well, it has to do with some physics, but we'll keep it simple and just say it mostly has to do with refraction. Refraction means that light bends whenever it passes through something or hits an object. So, as light passes through glass or water, it bends. And depending on what it passes through, it bends more or less. So when light in your microscope shines up through the glass slide, the light bends. So this is why we're going to get into why we need oil for our oil immersion lenses on our microscopes. So as you work through the first three objective lenses, 4x, 10x, 40x, specimens are easy enough to focus on. And part of that is because there's an adequate amount of light reaching your eyes from the specimen. But when you get to the oil immersion lens, you run into a problem. the hole on the lens is so tiny that normally not enough light from the specimen reaches your eye. So in the diagram, you can see that light is passing through the condenser lenses, and then they get to the slide. And then as they get to the slide, light is scattering away from the lens. That's refraction. Air is a terrible medium for light to pass through. Air causes light to scatter very severely. So if you're viewing your specimen under oil immersion, without oil, you could sit there all day long and focus, and you could not bring your specimens into focus. It would just look blurry because too much light is scattering away. So under oil immersion, we're supposed to add immersion oil to our lens. This is why. The oil causes the light to refract less. So as you can see with the blue line, as light hits the oil, it doesn't scatter away as severely as it does with the air. So oil is added to help more light from the specimen reach your eye. So when you add oil to the slide for oil immersion, you can focus on your specimen because more light is reaching your eye, allowing you to resolve those two close together tiny bacteria, or I guess 100 close together tiny bacteria. All right, so one other concept to address for what microscopes do for you. Contrast. So contrast is the number of visible shades in a specimen against a background. Okay, so that means, can you see an object against a background? That's simply what it is. So if you have a high contrast specimen, that means you have a very dark colored specimen against a very bright background. So imagine you have a black bacteria against a bright white background. That's very easy to see that dark black bacteria because there's a very severe difference in contrast. Low contrast would be colors of lighter shades against each other. So a gray object against a gray background, or a pastel yellow bacteria against a bright yellow background. So high contrast, big difference in color, black versus white, for instance. Low contrast would be more muted colors or similar colors against each other. So why are we talking about contrast here? Well, many microscopic specimens do vary in contrast. The problem is with bacteria, they have basically no contrast. They're essentially glass. They're nearly transparent. So if you were to put your specimens under the microscope, just naked, it would be very difficult to see. You would be focusing on glass. So we have to give our bacteria contrast artificially. So that's where we're going to get into staining. So we apply very dark colored stains to our bacteria to boost their contrast, to make them dark purple, dark blue, black, orange against a bright white background, so they're easier to see. And this is also why cells have to be dead. Typically, we have to kill our bacteria in order to see them, in order to stain them. So this is what we'll talk about more in lab, but a brief introduction into staining bacterial specimens. So like I said, bacteria are extremely low contrast. They're essentially glass. So we have to prepare them to be stained to boost their contrast. So in lab, we're going to learn how to set up smears. So how to apply bacteria to a slide, and then we're going to have to heat fix them. And heat fixing is a double-edged sword. It will help prepare the bacteria for staining. It helps them stick to the glass, but it also kills them in the process. But once you have your cells heat fixed to the glass, there's all sorts of staining techniques out there you can utilize. So in lab, we're going to learn how to do simple stains. So we're going to apply one dark colored stain to our bacteria. And then simple stains doesn't tell us too much about our bacteria. It gives you basic morphological characteristics. So are they round bacteria? Are they rod-shaped? Are they corkscrew-shaped? How many bacteria are there? Just very basic stuff. We're also going to get into differential staining, which is a bit more of an advanced technique because you'll utilize multiple stains in order to break bacteria into different groups. So, for instance, we're going to do gram staining. In gram staining, gram-positive bacteria stain purple, while gram-negative bacteria stain pink. So, you do the same procedure for gram-positive or gram-negative, but in the end, because you utilize multiple stains, you get two different colored outcomes. If you get purple cells, you know they're gram-positive. If they're pink cells, you know they're gram-negative. And we'll see a few other of these differential stains in lab as well. Negative stains, we only get to see once. They're pretty rare. Here you'd stain the background of the slide instead of staining the bacteria. So, smear preparation, we'll get to do this in lab soon, is very easy. You apply wet bacteria to a slide. You allow them to completely air dry, so you get this nice crust of bacteria on the glass slide. You then heat fix by passing your slide through the Bunsen burner. And once they're heat fixed and the cells are dead and cooked to the slide, then you can do whatever staining procedure you want so that you can see your bacteria under the microscope. So, some examples of stains that we'll get to see in lab. We'll get to do gram staining in lab. So you can see in the bottom left-hand corner, you've got purple cells that are gram-positive. And then you've got a bunch of pink cells that are gram-negative. Another one we'll get to do in lab is in the upper right-hand corner, is endospore staining. So here, we're differentiating bacteria that can make endospores from those that can't. So the little green eggs are endospores. The red chains are vegetative cells that are still active. The other two examples we don't get to see here at Harper, unfortunately, because the chemicals involved are fairly hazardous. Upper left-hand corner, you have capsule staining. This is a type of negative stain, so you can see the background is stained pink. And then surrounding the cells, you have that kind of clear bubble. That clear bubble is what's called a capsule. This is an important stain for identifying pathogens, as over 60% of pathogenic bacteria make capsules. And then the bottom right-hand corner is a flagella stain. I've never done a flagella stain. I've always wanted to. It involves pretty hazardous chemicals, but it allows you to determine whether or not your bacteria make flagella and what sort of arrangement. So you can see on either end you have flagella. Normally these would be too thin to see, but this particular staining technique allows you to thicken the flagella by basically just caking them with stain until you can actually see them. Alright, so what if you don't want to kill your specimen? Right? What if you have a low contrast specimen that has features inside of it that you want to see? Because if you do a normal specimen where we go through some sort of staining procedure, one, you're going to have to kill the cell, and two, by staining it, you're going to ruin these little features. You won't be able to see them. So this is where I want to introduce you to some alternative light microscopes. Okay, so these are just different takes on bright field microscopes, or some of them are just totally different altogether, but they are light microscopes. that allow you to see specimens without killing them so that you can see finer low contrast features. So the first example is a phase contrast microscope. Many bright field microscopes come with this feature built in. All this is a filter, and the filter amplifies the refractive index, meaning it acts kind of like immersion oil. It increases how much light from the specimen gets to your eye, and it will actually filter out some of the background light. So more light from the specimen gets to your eye, more background light gets filtered out, so you heavily boost the contrast of your specimen. So you can see in the picture of the algae in the upper right, you can make out many fine features inside of that particular algal cell. You can see the little vacuoles, you can see the chloroplast. If you had stained this specimen, you wouldn't be able to see that. Now, another variation on light microscopes are differential interference contrast scopes, DIC scopes. I've seen these as well. They're pretty neat, they're fairly large, and I have a picture of one right here. So this microscope is a little different than our scope because it utilizes multiple prisms. And folks, if you've never played with a prism, prisms are pieces of glass that allow you to split beams of light. So in this microscope, you have a prism above the specimen and a prism below the specimen. So the idea is that the first prism splits the beam of light, so you hit the specimen with two beams of light, and then those two beams of light are then recollected by the top prism. By doing this, by hitting your specimen with these two beams of light and then recollecting them, you create these beautiful three-dimensional images. So you can see in the bottom right-hand corner, you have some kind of specimen. I'm not sure what it is, unfortunately, but those fine glass features are only visible because of the use of this DIC microscope. If you had stained this or used some other light microscope, you wouldn't be able to see those fine features. Another one that's not particularly common, but still fairly commonly utilized, is dark field microscopes. So in the image, you can see that the background is black and the specimen is brightly illuminated. So here it becomes a trick of how do you eliminate the background light while still getting light from the specimen to your eyes? And it's actually a very easy trick. Instead of hitting the specimen head on with a beam of light, like our microscopes, you hit the specimen at an angle. So this is just like, and I'm sure all of you have experienced this, if you're sitting in a room and you can see the dust floating around in the air, right, because there's a beam of light kind of passing through from a window, you can only see that dust floating in the beam of light because light from the dust is reaching your eyes, while the light from the sun is not. So this works on the same principle. Okay, light hits the specimen at an angle, so only the light bouncing off the specimen gets to your eye. Any light from the background is eliminated. And I've also included a video for you here to watch if you want to learn a little bit more. All right, now, fluorescence microscopes are becoming just ridiculously common. You can specialize this as a career choice if you want. But fluorescence microscopes rely on ultraviolet light. So instead of a standard light bulb, you rely on a UV light bulb. So in the image, the background's black, but the specimens are glowing. Background's black because your eyes cannot perceive ultraviolet light. But the specimens are glowing because they are reacting to the UV light. and they are producing visible light as a consequence. So some specimens are naturally fluorescent, others can be stained and still living, so that you can see those brightly glowing specimens against this black background. Confocal microscopes, never worked with them, never seen one, but apparently they're out there. These microscopes don't utilize light bulbs. Instead, they utilize laser beams. And the best way to think of them is like a Xerox machine or a scanner. They utilize laser beams to scan larger objects, creating three-dimensional images. These are being commonly used for imaging bacterial communities, such as biofilms, so that you can see what the overall three-dimensional structure of that community looks like. So in the bottom left-hand image there, that is a computer rendering of what a three-dimensional biofilm community looks like. And you can see a picture of the whole confocal microscope in the bottom right-hand side. All right, so let's talk electron microscopes. So these are daunting tools that produce some of our most vibrant images of very, very small objects. Now, while electron microscopes are very powerful and many of them take up an entire room, they actually work on principles nearly identical to your light microscopes, and I can show you. So, electron microscopes don't utilize a light bulb. They utilize an electron gun. So, light bulbs produce beams of photons. Electron guns produce beams of electrons. So, following along the diagram. So, in our light microscopes, the light from the bulb is collected by a condenser lens. In an electron microscope, the electrons are collected by an electromagnet. Works just like a lens. It focuses the beam of electrons. The light microscope photons bounce off the specimen. In the electron microscope, the electrons basically blast through the specimen. But in the light microscope, the light beam or the photons from the specimen that bounce off of them are collected by an objective lens. In an electron microscope, they're collected by yet another electromagnet that serves just the same principle as an objective lens. Eventually, the image gets flipped. It would normally pass through your ocular lens in your light microscope, but instead, in an electron microscope, you guessed it, yet another electromagnet. Now, with light microscopes, you can view the specimen with your own eyes. With electron microscopes, you don't get to do that. You do not want a beam of electrons shooting you in the eyeballs, so instead, you have to view it on a separate viewing screen. So, some other interesting things about electron microscopes. Electron microscopes only produce black and white images. And some of you may have seen just beautifully colored electron microscope images. I have bad news. It's all done with Photoshop after the fact. They're given high contrast colors so that you can see different objects within the image. And I'll show you examples here in a second. They're very powerful microscopes. They can magnify objects up to 100,000x versus our light microscopes, which only went to 1,000x. The resolving power is pretty ridiculous. Most of them can get down to 0.3 nanometers, which means they are particularly useful for visualizing viruses, and they are the only good way to visualize viruses, and they can allow us to see the insides of cells and make out internal cell structures. But the biggest drawback, specimens have to be dead. No exception to the rule here. Alright, so two major types of electron microscopes. You have transmission electron and scanning electron. So in transmission electron microscopes, it's basically what I just showed you in the previous diagram. You prepare a specimen in what looks like a small crystal. You take very, very thin sections of that crystal, and then you blast the specimen with an electron gun. So it becomes a question of how many electrons passed through the specimen. So over in the image on the right, it looks like this weird kind of long blob with what I think looks like nuts inside of it, or maybe pistachios. That is a cross-section of a bacterial cell. And the different shades of color inside are areas of varying density. So the areas of lighter shades are less dense. The darker areas are areas that are more dense. So what you're seeing is a density map. So the darker areas are areas where less electrons got through. The lighter areas, the lighter shades, are where more electrons got through. So it's a question of how many electrons got through these varying parts of the specimen. These are our most powerful electron microscopes. They range from 10,000 to 100,000 x magnification, and they allow us to see primarily the insides of cells and to get viral images. Now, if you've ever seen an electron microscope image, it probably came from a scanning electron microscope. They produce very beautiful images. They're a little different than transmission electron microscopes. They don't blast electrons through the specimen. Instead, they use electrons to scan the surface of a specimen. So these generate three-dimensional surface maps. They're good for looking at the surfaces of cells and some viruses. They're not as powerful as transmission electron microscopes. So you're looking at 100,000 to 10,000 x magnification. So two examples of a couple of famous scanning electron images. So we're looking at the surface of objects. Top left-hand corner, if you've never seen this before, is a helper T cell from a human. So the big green blob is your helper T cell. The little red objects, those are actually HIV viruses. And what you're watching is a viral infection of that helper T cell in real time, or a snapshot. The other image, though, you've got some brown rough object covered by little green rods. Well, the brown object is a piece of iron, and the little green objects are a type of bacteria called Geobacter. Geobacter bacteria love iron because they can break it down. You're watching bacteria rust iron by stripping it of its electrons. So, diagram here comparing transmission versus scanning electron microscopes. So you can see with transmission electron microscopes on the left-hand side, the electrons are blasting right through the specimen, versus scanning electron, where the electron gun's a little bit more gentle and it's just bouncing electrons off the specimen, getting that surface map. So, transmission electron allows you to see inside the cells, while scanning electron just allows you to map the surface of the cells. Alright, now, the last one, I promise, scanned probed microscopy. That's where we can talk about atomic force microscopes and scanning tunneling microscopes. So these, I use the term microscope loosely here. These are microscopes. They are incredibly powerful, but you typically find them in a chemistry or physics lab. And you can see in the image in the bottom right-hand side, that does not look like your typical microscope. So these guys are very powerful. They allow us to see atoms, molecules, and even more recently chemical bonds. And while they probably seem incredibly daunting, they actually work on a principle that's incredibly relatable. Most of you folks probably know what vinyl records are. You're like, Why is he talking about vinyl records in a microbiology class? Because these work just like vinyl record players. So, with a vinyl record, you have a record that has grooves with little bumps in them. And in order to generate sound from that groove, you have to drag a needle across the surface of that record. And as the record spins, the needle hits the bumps, and as it hits the bumps, it generates vibrations which create sound. Same idea here. In these microscopes, a needle made out of tungsten atoms is either dragged across the surface of a molecule, or it uses protons to scan the surface of that molecule. So, by using this atomic needle, you can generate a surface image of an atom. So, a couple of fun pictures here. On the left-hand side, we've got a picture of a string of proteins. In the middle, we have some circular protein structures. But on the right-hand side, we've got one of my favorites. This is a very big Nature article that came out a couple years ago, where in the top image on the right, that's a three-dimensional rendering by a computer of what this molecule should look like. The bottom image on the right, though, is an atomic force image taken of that same molecule. So here we can prove that the computer model's right. That's what the structure looks like, and you can also see the chemical bonds. All right, folks, so that's it for Chapter 3.